Assalamu alaikum everyone. Today in this video lecture we are going to discuss enzyme and their classification. So basically enzymes are thermolabile biocatalysts which speed up chemical reactions without themselves being used up. It means enzymes are protein in nature. Enzymes are thermolabile. It means they are heat sensitive. When we provide high heat or temperature, enzymes are denatured because they are proteins. So proteins at high temperature denatured. And biocatalyst means they are catalysts that speed up chemical reactions in living bodies. If this is a chemical reaction which is occurring in living bodies, then the catalysts used are called biocatalysts, and they are not used up in a chemical reaction because enzymes only speed up a chemical reaction and they are recovered at the end of a reaction, and they can be used again and again in chemical reactions which are occurring in living bodies. Here is classification of enzymes. Enzymes are classified into 6 classes. Recently 7th one is added as well. Here is a mnemonic to memorize all classes of enzyme, that is OTHLILT. It means O, over the hill, life is lovely, terrific. O stands for Oxidoreductases, T for Transferases, H for Hydrolases, L for Lyases, I for Isomerases, L for Ligases, T for Translocases. So these are the classes of enzymes. In which class enzymes are classified? The first one is Oxidoreductases, Transferases, Hydrolases, Lyases, Isomerases, Ligases, and Translocases. So we will discuss each class in detail to get it. The first class of enzyme, that is Oxidoreductases. The name itself shows oxido means oxidation, reductase means reduction, ase, and we know AAC suffix, it shows enzymes. So these are enzymes which catalyze oxidation of one substrate with simultaneous reduction of another substrate. The examples are lactate dehydrogenase, succinate dehydrogenase. Here is general reaction for oxidoreductases. The first substrate is in reduced form AH because having hydrogen. Reduction is gain of hydrogen, gain of electron, loss of oxygen, while oxidation is gain of oxygen, loss of electron, loss of hydrogen. Here the first substrate is in reduced form. It is being oxidized because of removal of hydrogen, while other one is in oxidized form. It is being reduced by the addition of hydrogen. Here the first example through reaction, that is lactate. Lactate is being converted into pyruvate. It means lactate is in reduced form, it was oxidized into pyruvate, and here the loss of hydrogen that occurred is accepted by NAD plus and it is being converted into NADH. It means there is simultaneous oxidation and reduction of substrates. Next example, that is succinate. Succinate is in reduced form. It is oxidized into fumarate, and the hydrogen lost due to oxidation was accepted by FAD plus and has been converted into FADH2. It means there is oxidation of succinate and reduction of FAD plus. Next classes of transferases. The name itself shows that they transfer a group other than hydrogen from one substrate to another substrate, and the group being transferred may be any methyl group, amino group, phosphate group, or carbon dioxide, anything else. Here are the examples: kinase and transaminase or aminotransferases. Kinase often transfer phosphate group, keep in mind, and transaminase, it is second class. So here is general reaction for transferases. A and BX both are substrate. They are interacted in the presence of an enzyme transferase and they are converted into product. So here X is a group other than hydrogen. It is being transferred from B substrate into A substrate. Here is the reaction. It means there is a transfer of a group other than hydrogen. Here are examples: glucose. Glucose in the presence of hexokinase gets, here hexokinase transfers the phosphate group from ATP into glucose, hence glucose is being converted into glucose 6-phosphate and ATP is converted into ADP. Lost phosphate was transferred into glucose. Next is Next examples of alanine plus alpha ketoglutarate. One is amino acid and this one is keto acid. Both are interacted in the presence of enzyme, that is alanine transaminase. Here coenzyme pyridoxal phosphate is used and amino group was transferred. Hence the resultant compounds are pyruvate plus glutamate. Third class of enzymes, that is hydrolases. They are enzymes which catalyze hydrolytic reactions. It means these are the enzymes which hydrolyze ester, ether, peptide or glycosidic bonds by addition of water and breaking the bonds. It means by the addition of water the bonds are broken down in these cases. Here are examples. All digestive enzymes are hydrolases like lipases, proteases, amylases, etc. and urease. Here is example of sucrose, which is glucose. It is disaccharide. It is being converted into glucose plus fructose. And here the enzyme, that is sucrase, in the presence or by the addition of water, the sucrose is converted into glucose plus fructose. Next examples of urea. Urea is being converted into ammonia plus carbon dioxide in the presence of an enzyme urease, and here water is added. Fourth class of enzymes, that is lyases. They are the enzymes that catalyze non-hydrolytic reactions. It means they are just opposite to hydrolases. In hydrolases we provide water to break the bonds. Here we break the bond but we do not provide water. So we can say lyases are enzymes which catalyze splitting of various compounds by mechanisms other than hydrolysis. Here are examples: aldolase and second one is argininosuccinase. Let's see the example where aldolase is used. Fructose 1,6-bisphosphate. It is 6 carbon containing compound in glycolysis. It is converted or broken down into 2-3 carbon containing compounds, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate in the presence of enzyme, that is aldolase. Here we do not provide water because it is lyases class. Next example of lyases, that is argininosuccinate. It is being converted into arginine and fumarate in the presence of an enzyme, that is argininosuccinase. Fifth class of enzymes, that is isomerases. Isomerases are the enzymes which catalyze interconversion of isomers, and isomers are the compounds which have same molecular formula but different structural formula. Like glucose and fructose, both are isomers of each other and they have same molecular formula like C6H12O6, but their structural formula are different, as glucose contains aldehyde group while fructose contains ketone group. Here are examples: phosphohexose isomerase, second one is triose phosphate isomerase. Let's see the first example. Here glucose 6-phosphate, that is aldohexose, it is converted into its isomer, that is fructose 6-phosphate, which is a ketohexose. Here we provide the enzyme, that is phosphohexose isomerase. It means 6 carbon containing compound having phosphate, it is converted into its isomer. Next examples of glyceraldehyde 3-phosphate. It is 3 carbon containing aldotriose. It is being converted into its isomer, that is dihydroxyacetone phosphate. Sixth class of enzymes, that is ligases. Ligases are the enzymes that catalyze reactions where two molecules are joined together and ATP is used in this reaction. Examples are acetyl coenzyme carboxylase and glutamine synthetase. Here are the examples of ligases. Like acetyl coenzyme, 2 carbon containing compound, it was reacted with carbon dioxide in the presence of ATP, and the enzyme we provided, that is acetyl coenzyme carboxylase. As a result, malonyl coenzyme, 3 carbon containing compound is formed. Here 2 compounds are joined together using ATP. And keep in mind that there are 2 kinds of enzymes. One is synthase enzyme and one is synthetase. Synthase do not need any ATP to proceed the reaction, while synthetase enzymes are those enzymes which need ATP for their completion. Here the example of glutamate. that was added with substrate ammonia as in the presence of enzyme that is glutamine synthetase, and ATP was provided. As a result, glutamine was formed. Last but not the least, seventh class of enzyme, that is translocases. Translocases are the enzymes that catalyze movement of ions or molecules. Molecules may be carbohydrates, proteins, etc. across membranes or their separation within the membrane. It means they are enzymes that control either the movement of molecules across the membranes, or they keep the you can say molecules separate within the membrane. And here are the examples, like proton pump hydrogen ions, and second example, that is carnitine acyl carnitine translocase. These are the enzymes that control the movement of carnitine, or the so it was all about the classification of enzyme in brief. If you have any confusion, do ask in comment section. Thank you.